Welcome to this lecture on parturition and hormonal regulation. Imagine you are baking a delicate cake in an oven. You do not want to take it out too early or it will be undercooked, but leaving it in too long means it will burn. The human body faces a very similar challenge when growing a baby. Today, we are going to explore the brilliant communication system the body uses to know exactly when the baking is done, ensuring the baby is born at the exact right time for survival. Now that we have our baking analogy in mind, let us define what this process actually is. Parturition is the scientific word for childbirth. It is not just a mechanical event, but a highly complex physiological and neuroendocrine process. The baby essentially sends a chemical text message to the mother saying its lungs are ready. The mother's body receives this message and begins a massive, powerful process to open the doors and gently push the baby into the world. This ensures the safe delivery of both the fully developed fetus and the placenta. Understanding what parturition is leads us to how it physically unfolds. Clinically, this entire process is characterized by three distinct stages. First, we have the dilation and thinning of the cervix, which creates the opening for the baby to pass through. Second is the active expulsion of the fetus, which is the actual delivery of the baby. Finally, the third stage is the expulsion of the placenta, which nourished the baby throughout the pregnancy. Each of these stages must happen in this exact order, and they are strictly governed by a cascade of hormonal shifts. So, what exactly starts this three-stage process? It actually begins with the baby. As the fetus matures, its hypothalamic pituitary adrenal axis activates. The fetal adrenal glands release a massive surge of a hormone called cortisol. This fetal cortisol acts as the primary trigger, altering the steroid hormone balance within the mother's placenta. It effectively flips a biological switch, shifting the mother's body from a state of maintaining the pregnancy to a state of active labor. This crucial shift then triggers local prostaglandins and systemic oxytocin to soften the cervical tissue. This delicate hormonal timeline we just discussed is absolutely critical in the fields of obstetrics and reproductive medicine. If this hormonal cascade starts too early, it leads to preterm labor. This is dangerous because the infant's organs, especially their fragile lungs, are underdeveloped. Conversely, if the process fails to start naturally, post-term pregnancies can result in severe fetal distress as the aging placenta fails to provide enough oxygen. In real-world clinical settings, doctors can literally save lives by using synthetic versions of these exact hormones to artificially induce or augment labor when necessary. To understand how doctors manipulate these hormones, we first need to look at the natural balance of two major steroid hormones, estrogen and progesterone. Throughout most of the pregnancy, progesterone levels are incredibly high. This acts as a biological block that keeps the uterine muscles completely relaxed, preventing premature birth. However, as parturition approaches, that fetal cortisol we mentioned earlier causes enzymes in the placenta to convert this progesterone into estrogen. This removal of the progesterone block is what allows the uterus to transition from a quiet state to a highly active, contracting state. This shift from progesterone to estrogen can be represented mathematically. We call this the uterine activation index, denoted by the variable A. The formula is A equals the concentration of estrogen, E, divided by the concentration of progesterone, P. Conceptually, this ratio tells us how sensitive the uterus is to contraction-inducing signals. Estrogen is typically measured in picograms per milliliter, and progesterone in nanograms per milliliter. When progesterone is very high, the denominator is large, making the overall ratio A very small, meaning the uterus is relaxed. But as estrogen increases and progesterone decreases, this ratio skyrockets. Now that we have our formula, let us see this with a concrete example. A pregnant patient is at 38 weeks of gestation. Her blood tests reveal a serum estrogen concentration of 15,000 picograms per milliliter and a progesterone concentration of 150 nanograms per milliliter. We are asked to calculate the activation index to check for uterine readiness. We choose our formula, A equals E over P, and substitute our given values. Dividing 15,000 by 150 gives us an initial activation index of 100. Physically, this represents the baseline quiet state of the uterus before the final labor cascade begins. Following up on our patient, let us look at her numbers 1 week later, at 39 weeks. Her estrogen has now risen to 24,000, and her progesterone has dropped to 80. We are asked to find the new activation index and compare it. We again substitute these new values into our formula, dividing 24,000 by 80. This gives us a new activation index of 300. The index has jumped from 100 to 300. This massive threefold increase physically means the progesterone block has been removed, and the patient's body is hormonally prepared to begin active labor very soon. This massive threefold increase in our activation index does something very specific to the anatomy of the uterus. The high estrogen ratio physically stimulates the uterine muscle cells to change. First, it promotes the formation of gap junctions between the cells, which act like communication bridges, allowing electrical signals to pass instantly across the tissue. Second, it drastically increases the number of oxytocin receptors on the surface of these cells. Together, these changes perfectly prime the uterus, transforming it from a relaxed pouch into a highly coordinated muscle ready for powerful, synchronized contractions. Once the uterus is primed with all those new receptors, it is ready for the main event, which is driven by a hormone called oxytocin. Oxytocin operates on a fascinating biological principle called a positive feedback loop, specifically known as the Ferguson reflex. Here's how it works. When the baby's head pushes against the cervix, sensory nerve impulses travel up to the mother's brain. The brain responds by releasing oxytocin into the bloodstream. This oxytocin binds to those new receptors, making the uterus contract harder. This pushes the baby's head even harder against the cervix, which releases even more oxytocin, creating a continuous amplifying cycle. Because this positive feedback loop continuously amplifies itself, we can model the force of these uterine contractions over time as an exponential growth function. The formula is F of t equals F naught times E raised to the power of K times the concentration of oxytocin. Let us break this down. F of t is the contraction force at any given time. F naught is the initial baseline resting tone of the uterus before the loop fully engages. The constant E represents continuous compounding growth. The variable K is a sensitivity constant, depending on how many receptors the uterus built up earlier. Finally, OX is the oxytocin concentration. Because oxytocin is in the exponent, even a small increase leads to a massive, exponential increase in contraction force. Let us apply this exponential model to a real clinical scenario. During a medical induction of labor, a patient is given synthetic oxytocin. We are given a baseline contraction force of 20 millimeters of mercury, a sensitivity constant of 0.1, and an oxytocin concentration of 10 milliunits per minute. We need to calculate the new contraction force. We choose our exponential growth formula and plug in our values. In the exponent, multiplying 0.1 by 10 gives us exactly 1. The equation simplifies to 20 multiplied by Euler's number, which is approximately 2.718. Multiplying these gives a final force of 54.36 millimeters of mercury. Physically, this means the oxytocin has more than doubled the resting tone, pushing the uterus well into the active labor threshold. Now that we have calculated the forces involved, let us visualize this entire positive feedback loop in action. In this diagram, you can see the complete cycle of the Ferguson reflex. Starting at the bottom, the baby's head applies pressure to the cervix. Follow the arrow on the left. This pressure sends sensory nerve impulses straight up to the mother's hypothalamus and posterior pituitary gland in the brain. Moving to the right, the brain responds by releasing oxytocin into the bloodstream, which travels back down to the uterus. This causes the uterine walls to contract, pushing the baby down, which starts the cycle all over again. This diagram perfectly illustrates how the body uses a self-amplifying loop to generate the immense power needed for delivery. We have covered a lot of ground today, so let us summarize the most important ideas. First, remember that parturition is a masterclass in biological timing, initiated by the baby itself through a fetal cortisol surge. Second, this cortisol is the key that flips the maternal hormonal switch, removing the relaxing progesterone block and establishing estrogen dominance. Third, this estrogen primes the uterus, building the necessary communication junctions and receptors. Finally, oxytocin takes over, driving the powerful, self-amplifying Ferguson reflex that transforms the uterus into an engine of delivery. You now have a solid understanding of the hormonal regulation behind childbirth.